Turn this on and off. Hello? Yeah.
Hi, everyone. Welcome. I think I know most of you, but I'm not always a familiar face here. So my name is Leonard Foster, and I'm not going to be talking today apart from doing introductions. The people who will be talking are all from our group, though. We have, for those who don't know, we're sort of physically split, at least our mass spectrometers are. Our proteomics instruments are in the NCE building and Michael Smith Labs. And the metabolomics are over here in the LSI. And we develop and apply mostly quantitative methods originally mostly in proteomics, but we've started moving a lot into lipidomics as well. Sorry, not just lipidomics, but also metabolomics more generally. So today you're going to hear from three people in the group that are all doing sort of linked things, and I think you'll see how they all fit together here. Their names are all up on the screen there. So Moore, Lucy, and Juan are in our group. As I said, Moore and Lucy are grad students, PhD students in the biochem program, and Juan is a postdoc. Moore has been developing automation methods for more sort of high throughput and more efficient sample processing of larger cohorts of data. Lucy has been developing single cell proteomics and more recently single cell lipidomics and metabolomics methods. And then Juan is entirely dry bench work focused on multi-omics analyses and data imputation for big data. And hopefully you'll see how it all ties together. So I'm going to let them come up and talk. They've planned transitions. I will come back and manage questions at the end. So thanks very much for coming.
Hi, everyone. Thanks for coming. So I will start it off. First of all, just thanking the lab and the proteomics and metabolomics core facility, who obviously run all our mass spectrometers and where all this data was processed from. So at the top left is the MSL building, where we have our proteomics core facility managed by Jason at the top right. And in this building on the second floor, we have our metabolomics core with Armando, who's at the bottom right there. So this here is just kind of an overview of our three talks. So I'm mainly going to be talking about bulk multiomics and how mass spectrometry is used for that. Then it'll transition into Lucy talking about single cell multiomics, kind of looking at those subpopulation differences that are sometimes masked when you're looking at the bulk samples. And then it'll finish off with Juan going more into detail about the bioinformatics that we need to use, specifically when we're integrating multiomic data, how we do it, and what we need to do differently between the bulk and the single cell methods. So most of the data that I'm going to talk about today is from the Canadian Microbiome Mapping Initiative. Briefly here, this is a multi-group collaboration with all different labs here in the LSI and the Michael Smith Lab. The idea is to kind of develop this multiomics pipeline where we can take participants from different studies and then run them through with different sample types, do all the different sample preps. So mainly we're doing the mass spec, so proteomics, lipidomics, metabolomics, but other ones in this group that are working with us are doing epigenetics, genomics, transcriptomics, microbiome work, and then figure out how we can first of all process those individually, but then integrate the data in a meaningful way to be able to help clinicians at the end of the day, and then the patients especially. So when we're working with large scale studies, there's a lot of words on the board, but these here are just different considerations. And my talk's going to focus on these three. So I'll start out by talking a little bit about sample preparation and enrichment strategies. Then I'll touch on multiomics integration, kind of from the data output end, and then talk about how we really need to change the way that we do things to allow large scale clinical studies on the throughput end. So first of all, everybody wants to get the most of their data. So sample prep is really about how do we get the best data from the samples that we're given. So we can start with a crappy example of some fecal samples. And if this here is the first time that a lot of you guys have seen a chromatogram, that's basically our visual output from our mass spectrometer. What we're hoping for is a bunch of peaks. Each of those peaks represent a peptide, and from those peptides, we're able to infer the proteins that are present in our sample. This one here is pretty bad. The reason being is that we don't have many peaks, and the ones that are there are very big and kind of overshadow a lot of the data. And this here, as you can imagine in fecal samples, there's not just proteins in there. So we had to think of a way to kind of help our downstream analyses by cleaning up the sample. And one of the ways that we can do that is actually quite beneficial for multiomics. It's called methyl terp bute ether extraction. So what this ether does, it's a multi-phase extraction where we get an organic layer on the top that contains the lipids of our samples. On the bottom there, we have an aqueous layer that's mainly our metabolites, and you actually get a nice pellet at the bottom that are quite clean proteins. And as you can see on the chromatogram on the right, we now have this huge influx of a whole bunch of thin peaks, one after the other. And thus, from that data, we actually get a lot more of a comprehensive proteome coverage. And when we're talking about proteomics, we really can only find stuff that we're looking for. Even if it's there, if we didn't look for it in our database search, we're never going to find it. And if you think about a metaproteomics experiment, what can be in these fecal samples? Do we only include human proteins, the microbiome, dietary proteins, or do we include everything? Well, computational limits really restrict it being everything, so we need to be a little selective here. So for the data that I'm showing today, basically we took human proteins. SwissProt is basically this large combination of all different model organisms and stuff that are in this curated database that's well annotated. We took common microbial species from the human gut, from healthy people, from previous literature, as well as information on the proteomes from the most abundant bacterial species based on transcriptomics of the same samples, as well as some realistic foods. And then highlighted at the bottom that I'll get to in another slide is some metatranscriptomic sequences from Avril at the Finley lab on the same sample. So this here is the actual sequence that they got in the transcripts. And we are looking for the corresponding peptides to see how these layers match up with one another, kind of hinting on why multiomics is important. And the reason we're now able to do these bigger database searches is from a new software that is really helping us with this. And just from this here, we're already around 13,000 unique proteins just from these 8 samples that we ran through the test. And for multiomics, the real question is, does the proteomics and the transcriptomics line up, or whatever other layer? But I'm going to use this here to kind of show the story. So first of all, when you look at the correlation, it's quite expected that they don't correlate well with one another. But this here is kind of my favorite graph for showing that. So the circle on the right, all those features are on the axes that represent 0 log fold change for the RNA. However, you're seeing huge changes at the protein level. And on the other axes, the x-axis there, you have stuff that isn't changing at the protein level, but is changing quite big at the RNA level. And this here is the reason that multiomics is starting to become a thing and is so important, is that a lot of studies might only be looking at one layer and be missing important biological information that they're just not tapping into. Another thing that we need to be conscious about when we're doing our sample prep for proteomics is basically the dynamic range. So this here is the dynamic range of proteins within blood. As we can expect, albumin, immunoglobulin, very concentrated in our blood, and a lot of the super important things in our blood for signaling, like interleukins, are way down at the bottom. So that's about 12 orders of dynamic range. And those top 20 proteins are about 99% of the total weight. So being able to see those ones are very hard when our instrument has a dynamic range of 10 to the 5. And whatever's biggest is taking up that range. So you kind of see that classical plasma protein end. But we want to be able to see the rest of that. And some ways that we can do this is with enrichment. So this here from that same group that we got the fecal samples, this here is urine and serum that were enriched. And basically comparing UH would be the urine and SS is serum. It's unique proteins and the overlapping ones. And the reason that we're getting so many more in urine as opposed to serum is the differences in this dynamic range. So what this curve kind of displays is that 5-fold change of the dynamic range. And the more that fit in that range, the more proteins we're able to find, such as the yellow. So when we zoom in there, we can see the blue line, which is serum, and plasma works very similarly. There's a lot of these super intense signals, and then it kind of dips underneath the yellow line and then goes across. So more proteins are showing up in this middle of the dynamic range. We're basically trying to compress it. So what these enrichment strategies do is try to reduce some of the high abundance things in our sample while trying to capture as much of the low abundance we can to kind of shrink that range and then end up with a better proteome coverage. So we have some of our data. The next step is how do we properly integrate it? So just to zoom out again, what I'm going to show you is protein, lipid, and metabolite data from urine. So that same data from previously using helic enrichment for the proteins, and then that extraction protocol for the multi-phases for the metabolites and the lipids. And as you can think, the first way that you can look at multiomics data is to shove it all into one Excel file and run your statistical analyses. And you get something like this, which is kind of fun. So some stuff that we can see just at a high level, we can separate out through clustering the male and female samples. We can see that some of those female samples are kind of in a distinct cluster from some of the others. On our PCA plot, which basically is showing the variation between samples, we're only at about 20% on PC1, so the data is quite complex. And we are getting a little bit of a separation between these two clusters. However, we lose a lot of the specific information to the layers. It's kind of hard to see, but a lot of, I'd say probably 80% of these show L underscore. So this is almost all lipids that we're looking at right now. And we're not really seeing the modality specific stuff. So if I did the same thing, but only looked at one layer at a time, it looks quite different. So we have, for the proteomics, a whole bunch of female samples with very high levels of a bunch of proteins, and then all the rest are kind of clustering together. Lipids kind of have a similar effect where we're not quite getting good male and female clustering, and we're starting to see all these differences. And then the one that seems like it shouldn't cluster well, metabolomics, is actually the driving force here for being able to tell between the male and female clusters. And we can also see on the metabolome that there's a lot more patient-specific differences even within the groups. So that's why it's not quite as clear-cut. But what we want to do is have this information from the individual layers, but a little bit more representative where we can combine it together. Just going into the PCA plot, we can see they all are able to cluster in their own way, but it's still not quite enough for being able to get a good information of our cross-modality comparisons. So is there a better way? One of the ways is using unsupervised integration models. So the idea here is to capture these complex differences, interactions, cross-modality interactions in a way that we can interpret. And unsupervised models have this unbiased aspect where they're looking for trends in the data and then you're labeling it. So you're basically looking at what the model has picked out as being important and then seeing where it came from. So I'm going to be talking about MOFA, which is a multiomics factor analysis. If you've ever worked with principal components, it's the same idea. Basically, these factors represent the main drivers of variation. So when we make our model, first thing we want to see is how do these factors line up with our metadata. So this here is just kind of example where we have association with both age and sex. And you might be a little hard to see, but there is a star there. So it was significantly associated in factor 1 being age. And we can see it's a bit darker along the top for the sex column for both factors 2 and 3, showing that the difference explained by those factors is now more attributed to sex than it is age. So what does this mean? Well, it means that we can then look at the individual features of the lipids, metabolites, and proteins that drive these factors, and they are then going to match up with being either more sex or age related. This here was just our trial cohort. Later this summer, we're going to be going up to around 220 participants and then being able to really expand this, not just with urine, but our other sample types. And something that is also kind of cool to note is that some of these features might be specific to one factor, but for example, here, you have a particular metabolite, for example, that is a driving force in 2 different factors. So maybe it has multiple roles and something that we'll be able to look at the larger cohort. And really, that comes to the next steps of this project, is that when we don't just have urine, we're going to have plasma, we're going to have fecal samples with all 3 omics. How do we put all that together to be able to see the main drivers of some of these differences? This here is just an 8-participant snapshot where we were just testing the whole pipeline, but hopefully in the next months, we'll be able to scale this up to the large set, which scaling up requires something that I can only do so fast. So that is why they invented robots, which we just got. This here's OpenTrons Flex. It's able to pipette whole plates at a time, much faster, and sadly more reproducible than me. So that there, along with a few other robots in the lab, we're really scaling this up to be able to reproducibly and with a high turnaround handle our large sample sets. So now I'll invite Lucy to talk about how single cell answers something that I don't.
All right, so I'm gonna shift gears a little bit and talk about the single cell work that we have in the lab. So the most motivation behind doing single cell work is this idea of cellular heterogeneities. So for example, if you take cells from the same tissue, such as the brain, the lung, or the muscle, you'll see a very diverse differences between the individual cells, even though they come from the same population. So in my case, I mainly focus on astrocytes, which is a type of brain cell that has many functions inside, including that they can maintain the blood-brain barrier formation, they interact with other cells inside the brain, they are part of the immune system of the brain, and so many other ones. So then this brings up the question of whether doing a bulk analysis by pulling them all together is going to mask that cellular heterogeneities of astrocytes. So to understand this, about 5 years ago, researchers started doing single cell level analysis on astrocytes. And at the time, the readily available tool is single cell RNA sequencing. So by using this, this particular research group identified 5 clusters of astrocytes. And then they concluded that this is the reason why astrocytes can have so many functions. But as we know that RNA and protein have very bad correlations, so then it brings up the question of whether the genomic and transcriptomic level information is really enough to tell us the full story of biological heterogeneity. So this is when our lab moved on to see if we can find a way to study proteins on the single cell level. But because we're doing single cell level analysis, so the sample amount is very small from the beginning, which is why for when we try to establish and optimize the workflow, we're trying to really minimize the sample manipulation and reagent used, as well as decrease the working volume as much as we can. So this is the generic workflow that we have currently in the lab. We first dispense the single cells into 0.5 to 1 microliters of water in a 384 well plate. And then we'll add 1 microliters of trypsin, incubated for 4 hours for digestion from peptides to proteins. Then after that, we load everything onto liquid chromatography and then run it through mass spec for analysis. So with this robust workflow that we have in the lab, we've ran many different types of samples, including the boring ones like HeLa, A549, and HAC. We've also done primary cells such as PBMCs and primary astrocytes, which is the main focus of my work. But we also tried it with tissue samples such as human brains and honeybee brains. So we have so much data. How do we actually understand them and make sure that they're actually good data for biological analysis downstream? One of the biggest problems that single cell proteomics field has is that people are really just chasing numbers because it's a new tool. Everybody wants to say that I'm getting more IDs than the previous group. So today you see a publication of 3,000 IDs that they can find. Tomorrow you see 5,000. So what does that really mean? So then our group really wants to advocate the idea that data evaluation for SCP is really about the reproducibility, the quantitative depth, and biological relevance, rather than just an ID number. So I'll give you an example. Here is when I tested the whether or not using reduction and alkylation for sample prep is necessary for a single cell. as I've mentioned, that we're really trying to decrease sample manipulation. So I've tested, here is without any reduction in alkylation. This is with reduction in alkylation. The other ones are just reducing only, which we can probably ignore by now. It's really bad data. But so the completeness here basically shows how reproducible we can detect the features across all the samples that we have. So for example, 80 to 100% means that there are roughly about like 1,300 proteins that we can see across 80 to 100% of the cells that we analyzed. And by adding that reducing and alkalizing agent, we're decreasing that ID or the reproducibility. But then if we're to look at the peptide to protein ratio, especially looking at the yellow and the brown ones here, there really isn't much of a difference between with and without reduction in alkylation. Then what I want to emphasize is here, this graph here shows the number of features, or I guess number of proteins we can see depending on the conditions that we use. With reduction in alkylation, we are seeing a little bit of a decrease with the total ID as well as the reproducibility shown earlier. But you are actually identifying that these 500 proteins that are going to be missed if you don't actually do the reduction in alkylation. So like number really is not what we should be looking at now. And instead of that, we should more focus on the actual biology, whether the protein you're interested in does require this step moving forward. So another aspect of data evaluation is to look at the biological interpretation of the data. For any conventional proteomics approach, it's very common that people do differential analysis and then visualize it by volcano plot downstream, which is not going to work for single cell because it averaged out all the intensities within the conditions, which then masks the cellular heterogeneity. So I'll give you another example here. This is the data that I got from my collaborators where they did a differential analysis or differentiation of a neuronal cell. At 0-day differentiation, we see a nice cluster of the cells over here on the UMAP. As they differentiate, it then divided into two groups of cells. And you can validate it by looking at the neuronal markers where some of the proteins have intensities split into two different groups. But if you're to do a conventional mass spec or proteomics analysis, we can see that it really is not picking up all that heterogeneities that we would be expecting from a single cell analysis. So from here, I'll show you a little bit of the astrocyte data that we get. Overall, we're also getting about 5 clusters of astrocytes. And when we look at the particular proteins involved in each, we can then group that into particular functions that astrocytes are responsible for in the cell, which is promising. But we know that proteins don't really act alone inside the brain, and a lot of the times they interact with downstream molecules such as lipids and metabolites. So we moved on and see if we can study lipidomics at the single cell level. So this is with help from this amazing person in our lab, Armando, who's also sitting in the audience today. The workflow is very similar to proteomics, but instead of water, we are dispensing the cells into 3 microliters of organic liquid, and then we do a vortex in sonicate to extract the lipids, and then run it on liquid chromatography and mass spec for lipid analysis. So from here, we are getting, like these are the lipid classes that we get, which is generally what we're expecting from a biology, which is good. So we are seeing the common lipids using this workflow. So from here, then we move on and see if we can integrate the protein and lipid together to help us understand the biology a bit better. The easiest way to do is basically take my protein data, lipid data, do a computational integration downstream. But the biggest problem is because we took protein from one cell and lipid from the other cell, due to the cellular heterogeneities, then we start to question whether this integration is actually biologically meaningful. So to solve that, we wanted to see if we can find a way to extract protein and lipid from the exact same cell. And this is when we came up with the workflow, which I call it MAPLE. But the idea is we're going to drop the cell in 200 nanoliters water, Then we do a quick lipid extraction first, and then whatever remaining will be then processed for proteomics. Like I won't go into too much detail. Then after that, we do computational integration. So this is what I call the most important QC data. The y-axis here shows the recovery of protein-lipid interaction. The middle one is the true same cell lipid-protein pair. And then the gray one is when we are trying to mimic protein from one cell, lipid from a different cell. And we're seeing a general decrease in the percent recovery when you don't have that two omics from the same cell, which have proven that doing same cell multi-omics is actually important. Then taking a look at the actual features that we get, overall for proteomics, we're getting about 2,500 IDs from each individual cell and around average of 4 magnitude dynamic range for protein. And then for lipid, we're getting about 1,000 or so features. I will say that out of those, only 200 or so are actually annotated features. And then we're getting about 3 magnitude dynamic range. Then I wanted to see, because we're getting protein and lipid from the exact same cell, I wanted to see if having more protein from one cell means that you get more lipids from the same cell, which doesn't seem to be the case at all in this case. There's no correlations at all. And then the next question that we asked was, since we're extracting another omics from cell, is that going to affect our initial proteomics quality? So this is looking at, sorry about the label, but this whole cell is when we just do a regular SCP workflow for proteomics. Half cell is after extraction of lipids, the remaining one goes through proteomics analysis. We're seeing a really high Pearson correlation score here, which means that we're actually not losing much protein. And same with this one is comparing the subpopulation, subcellular localization of the proteins. Generally, we're seeing basically identical percentage. And then the last one here is showing the completeness, as I mentioned before, comparing the regular SAP workflow versus after lipid extraction. We are seeing a little bit of a decrease in terms of the reproducibility of the protein, but overall, I'd say it's not too bad. And then this is looking at the peptide level. We do see a little bit of a decrease, although it's relatively minor. So the y-axis here shows the protein sequence coverage. So how many precursors per protein you get. We do get a little bit of a drop when you extract the lipid out. And then the one on the left is looking at the CV change and the completeness change here. There's a little bit of a decrease in the completeness on the peptide level, but I will say that with the benefit of getting another omics out of single cell, the drawbacks is very minimal. So from here, we have now a well-established workflow. Going back to the biology, I'm trying to study astrocyte cellular heterogeneity. And as I've mentioned that they're super heterogeneous. Juan will tell you a little bit more about it, but with this tool, we're trying to explore how astrocytes can respond to viral infections at the single cell level. And I'll pass it down to
Okay, I'm Juan. Now I try to move into the bioinformatics part of the project, and specifically using like the MOFA, the multi-omics factor analysis. So what is the mean of the factor, or what's the mean of the variance? So you can imagine this is a paper, and if I just fold this, and if you just direct it through this direction, this is just like a circle. It's a two-dimensional things. But however, if I look at that, I turn the direction like that, it's a cylinder, right? maybe change a little bit of the direction. If you direct to see this is irregular, maybe a shape. I don't know what this means. It's not very regular shape. So that means for our biology omics data, so we have multi dimensions that indicate those features like proteins, genes, or metabolites, or the lipids. And however, the number of the samples is really limited. So then that causes a very well-known topic that means the curse of the dimensionality. Like the numbers of the sample, that's lacking of the power to explain the variance or the changes through for those features. Then how to tackle with those problems? Then we're thinking about like the dimension reduction. So that means the factor you can imagine this is a view that you just want to like capture the major variance or major interesting biological question you want to ask, whether this is a sex-driven, this is an age-driven, or this is a disease-driven problem. So then back to our biology question, the key problem is that we have many different types of the data from the same biological systems, like the genomics, transcriptomics, proteomics, epigenomics, or lipidomics, or the metabolomics. And also we have other modalities like the image data, we have some clinical data, we have some metadata, or you have some perturbation system to like the virus infection. Then we try to just, as I said, reduce the dimension. Otherwise, we are lacking of the statistical power to explain all the things we observe. So that means we will do the instead of just integrate all the features together, we try to capture the major directions we want to look into the data. So then after that, after the model is trained, we can look at some factor scores for each of the samples or the cells. So those scores can help you to separate some group or like track some disease progression or the infection related trajectories. And finally, what is they mean? What those factors mean? And we try to interpret each factor by asking like which feature contribute most strongly and whether those factors is really associated with the real biological and the clinical problem. So the overall workflow is like this. So we have like four steps of that. And here's the first example, I analyzed all the data from Mauro and Lucy. So Mauro has these very big data sets for the Parkinson's disease for the multi-omics. And in this case, our input data has 3 layers, like proteomics, lipidomics, and the metabolomics, as well as some covariates, sex, age. Later we will have some recruit some participants who have the Parkinson's disease diagnosis. So they have the disease status. Then as we said, we first try to reduce the dimension into 3. Then we have 3 views to look at into the data sets. And then after that, do some interpretation. So for instance, maybe there are some factors that demonstrate like the disease status. Maybe there are some factors that explain like the metabolic process, or maybe some factors just give you some a hint for this is a technical bias. So this is just for example. And Hans is an undergraduate student from the combined major from the computer science and statistics. And he's under my supervision for 1 year. He did a very good job for these data sets. So the Parkinson's disease, for this Parkinson's disease multi-omics data sets, we will first try to do sensitivity analysis. That means we will try to see how many factors we need to explain the data sets. We don't know that. So that means, so that's why we do the trial. And you can see that the green line denotes for the metabolomics. It really captured the most of the variance compared to the other omic layer. And probably that means it's more sensitive to the disease status or like the changes of the donors. And other than that, we are wondering that whether those factors have some association with each other. Because when we do the dimensional reduction, we need to make sure that each of the axes or each of the dimension, they're independent with each other. They are orthogonal to each other. So that means we will do the correlation analysis, and we found that actually there are no relationship with each other. That means we really captured some direct direction with for the data sets. And as we said, the more factors we have, more variance captured. But this is a good thing or not? The number, when we increase the number of the factors, we actually increase the complexity of the models. So this is not the thing we want in the machine learning part. We want less feature, higher predictivity ability, as well as like less complexity of the models. So we will choose a relative balance one. That's 3 factors will be the best. And Mauro explained a little bit for the previous slide, so I will just give some details of that. We will just want to show like the factor score can help us to plot a very good heat map on showing like urine samples clusters based on their variability. And besides of that, you can see on the right, the plots also show the top features weight for the factor 1 across different omic layer, also including like the urine lipidomics, fecal lipidomics, urine metabolomics, and fecal metabolomics, so the proteomics for both tissues. And also the axis, we have the both of the normalized weights, both the positive and the negative. That means they have positive contribution to the factors. The other is the negative contribution to the factors. So the plots will help us to answer that which factor drive the, which features drive the factors the most. So by examining these top features, we can just move from the statistical factor to a biological interpretable Parkinson disease signatures for the downstream biomarker discovery. And other than that, we will move from the bulk MOFA to the single MOFA that use the stem cell proteomics and the lipidomics as from the introduction from Lucy to study the virus-driven astrocyte state dynamics. So the key difference is that like each data points represent an individual cell rather than a bulk sample. So for each of the cells, we have the hair, the proteomic, and the lipidomics readout, along with those metadata, such as the donor and the infection status by the infection status, which is induced by the COVID-229E virus strain. So this really allows us to study to ask how infection changes cellular state at a much higher resolution. So on the left, I just put a demo figure to say we can integrate those stem cell multi-omics data as well as some metadata together and to organize the cell along a virus response progression from the baseline of very early antiviral status, and then even higher states, and then finally remodeling states of those astrocytes. And on the right, I just want to draw a hypothesis of biological story interpretation of that. Is that like the protein and the lipid together, they can capture the host, the virus and the host remodeling. For instance, we can detect those viral structural protein like the capsid, nuclear capsid, envelope membrane, or like different the spike protein. And then how they affect the host cell, whether the receptor on the host cell changed, and for the vehicles, and how their RNA, how their RNA release, and how they do the replication in the host cells, and about like the protein synthesis and how they affect the ER Golgi trafficking or the host defense signaling, or finally maybe some protein stasis stress or the reactive remodeling things. And the main advantage is that our MOFA can really help us to review all the heterogeneous infection response instead of just averaging all the cells into a bulk story. So for here, we use the stem cell profile to define the astrocyte response states across the donors. The main message is that the infection does not create a one uniform astrocyte response. Instead of that, cell distribute across multiple cellular states. Some states are closer to the baseline, some are not. Like they may have more stronger, they have stronger infection associated activity. So the UMAP show this is all the like the controls and the infected cell are now simply separate into two clean groups. Instead of that, they distribute across several states, suggesting like the astrocyte response to infection is a heterogeneous way. Also, we tested the state composition. They change after the infection and they have shift vary across different donors. And when we try to interpret these states, we summarize the protein and the lipid infection scores. And the expanded states tend to show higher protein and the lipid infection responsive score, which links like a shift in the cellular state composition to coordinate molecular change. So you can see the trajectory here. So the main takeaway is that like our stem cell multi-omics can reveal those donor-dependent astrocyte response states as well as their trajectory. And besides of that, after the global discovery, we want to try to look at what happened for those individual features in the infection analysis. So after identifying those states, when they ask like whether there are really some infection changes the relationship between the proteins and the specific lipids. So on the left, you can see the circular plot that shows alter the protein lipid feature pairs, and that pairs connect the astrocyte stress proteins, viral proteins, and some membrane related lipids. And also when we like look at like a global scale, global view of that, like we can see that there's some antiviral in the immune response that are correlated with some like PC and PE lipids program. So that the red color means the positive correlation between those protein and the lipid program. So that as we said about that, like the infection will not change only one layer of the features. Instead of that, they will rewrite the protein lipid program together. And finally, we want to just connect the viral signal to the host remodeling and the lipid context. So you can see from the left, we detect the viral proteins, including the spike or other kind of the structural protein. And then the diagram links the viral signals to the entry and the trafficking host proteins, such as very well-known kerosene-related proteins. And then the proteins are connected to the downstream host modules, such as receptor-related entry and the vesicles and also the membrane trafficking. And after that, the next layer, I want to show some representative host protein here. The final, we want to link those protein modules programs with the lipids, such as some ceramide or PC balance, or some membrane phospholipid. So the main point is that the infection is visible at multiple levels, and we try to build a network for each among all the multi-omics data sets. So they really support the idea that the stem cell proteomics and the lipidomics can really capture the coordinate and some of these coordinate the viral host response. So I want to thank all the people. We also want to thank all the people coming here, and the most we want to thank our supervisor, Leonard, who gave us the chance here and to finish our very fantastic work here and provide very excellent, friendly environment and very supported infrastructure for our research. And also thank Armando, who is not present here, but he really contributes lots to our work. Yeah, thanks, Armando. And also thank Lucy and Mauro gave me very fantastic data sets to analyze. And yeah, thanks all.
All right, thanks guys for the talk. We've got a few minutes for introductions. Questions. Introductions are over. You guys can probably come up. Remember to use your mic if you can, please. Yeah, go ahead. Sorry.
Fantastic. So wonderful talks across the board. I had a question, I guess, for Lucy, but open, of course, to everybody. The single cell proteomics, metabolomics, lipidomics that you're showing, how close are we to pairing that with spatial approaches, not considering cost, which I'm sure would be substantial?
That is what I'm hoping to do, but Leonard's not letting me do it because I need to graduate. So that answers the question, hopefully. But like, you definitely can, like there are techniques out there that can do spatial level, not exactly single cell, but close enough to single cell that can provide that spatial layer information.
Maybe one thing to add to that is that the methods that people use for spatial omics at the transcript level don't really work for proteomics so well. In fact, most of the methods for single cell RNA-seq overall don't work or are not compatible with a mass spectrometer as a detector. So sort of having to reinvent that wheel. But one other thing that none of these guys talked about, but that Armando has got working and that we're excited about exploring as well is using mass spectrometry to image slices. So it's not quite spatial in the way you're thinking, but it's still, at least I think that you're thinking, but it's still spatial in that you can do an image of a cryosection slice.
Thank you for the talk. I have a really naive question for the first talk. Just wondering how much urine do you need to in order sufficient to do the process? Thank you.
So you don't need much to do it. To do it well and to be able to do the enrichment that I was talking about, it's about 300 microliters for the proteomics. And 200 microliters for the metabolomics and lipidomics together, and you have a lot of excess. But the reason the values are a little higher is just for being able to do enrichment techniques. From a few microliters, we could get results as well.
So to give you an idea of the dynamic range that you just heard about here today, Lucy's single cell proteomics is measuring probably about 250 picograms of protein per cell. And plasma has a concentration range of about 60 milligrams per milliliter. So 300 microliters of that is, what, anyone can do that math? 10 orders of magnitude more than what Lucy's measuring. So it's not a sensitivity issue, it's a sample handling issue. Yeah, go for it.
I got a bunch of questions because I thought it was an interesting talk. I guess I'm going to kind of ad lib here a little bit. When Lucy had shown the decreased coverage following extracting of the lipids and that you're losing some amount of proteins, is that because you think they're coming out due to protein adhesion onto the well surface because they may partition into the organic fraction? And I guess, is there merit to understanding why that's happening further? Like, are there proteins that are very tightly bound to lipids that then you're partitioning out, or is it really just like protein absorption to the, plastics?
I think there's a little bit of both. We're still trying to figure out what the reason is. When I looked at just protein level comparison between just single or intact cell versus extracted, hydrophobicity of the cell or the proteins are very similar. The sizes of them are very similar. Like I'm not seeing any trend to tell me what is causing that. So my guess is that I'm not actually losing, like in terms of like even the number of proteins I recover is more or less the same. So most likely I'm losing a little bit of peptides that are very low abundant, and that caused indirectly a little bit of decrease in the protein quality. Yeah, but like the type of peptide is not super clear what they actually are.
All right. Thanks very much, everyone. Thanks for coming out. Before I thank the speakers one last time, just a reminder to take your garbage away with you and recycle or compost stuff. And again, thanks to Hugh and CBR for hosting, and thanks to Moore, Lucy, and Juan for their talks today. Very, very smooth, guys. Very well done.